So chairing the session is Darshana Kapri, who's a postdoc in our lab, and she's going to introduce the next speakers. Thank you, Darshana.
Thank you, Martin. We now move to immune system, and we have 3 beautiful talks lined up for educating us about the immune cell mitochondria. First up, we have Jack Devine, who is a research associate in the lab, who will talk to us about the molecular regulation of immune cell mitochondria. Welcome, Jack.
Thank you. So as Darshana said, I'm a research assistant in the lab, and I'm going to tell you about immune cell mitotypes and how they change across the age range. Oh, sorry. Is that better? No. So in a similar way that there are many domains to cell biology, such as cell division, differentiation, contraction, migration, etc. There are also many different domains to mitochondrial biology. There's the membrane potential, respiration and oxphos, reactive oxygen species emission, and morphology, fusion and fission dynamics, and many others. And mitochondria are multifunctional. So they do oxphos and ATP synthesis, which is what they're best known for. But they also do a range of other functions, such as regulating calcium homeostasis, amino acid metabolism, ROS emission, steroid biosynthesis, and neurotransmitter synthesis, and many more. And even when you just look at mitochondria in different cell types, they look completely different. So these are electron micrographs of mitochondria in different cell types. And you can see their morphology and ultrastructure is very different in different cell types, indicating that they're performing very different functions in different types of cells and tissues. Our lab published a study in 2021 investigating the mitochondrial phenotypes of different immune cell subtypes. And in this study, we collected the blood from 21 individuals and isolated the PBMC immune cells, and then used fluorescent activated cell sorting to isolate this immune cell subtypes. And we then profiled their mitochondria by measuring the electron transport chain complexes 1, 2, and 4, the citrate synthase, or the Krebs cycle enzyme citrate synthase, as well as the mtDNA copy number. And then using these measures, we found that the different immune cell subtypes have different mitochondrial phenotypes. So I'm showing here in the biplot, we're showing citrate synthase activity on the x-axis and mtDNA copy number on the y-axis. And you can see that the different immune cell types occupy different spaces on the plot. So for example, innate immune cells and naive lymphocytes, they both have this low citrate synthase activity, but they differ hugely in mtDNA copy number. And this study was when our lab first started to think about the concept of mitotypes. So what are mitotypes? So mitochondria are multifunctional. They're not just a powerhouse. They dynamically respond to cellular and organismal needs, and they differ between tissues, cell types, and even subcellular compartments in their specific molecular features, activities, functions, and behaviors. So they specialize into distinct mitochondrial phenotypes, which we call mitotypes. And we're still lacking a systematic description of mitotypes. So to address this, Anna Monzel in our lab has developed a computational approach to quantifying mitochondrial specialization using omics datasets. So we can take single cell or bulk RNA-seq transcriptomics datasets or proteomics. And with the method, we filter the datasets for the mitochondrial genes. So we use the Mitocarda 3.0. It contains an inventory of all mitochondrial protein coding genes, of which there are 1,136. And mitochondria also provide they also provide 149 pathway annotations based on these genes, so which correspond to specific mitochondrial functions. So we can then go from the mitochondrial genes to 149 different specific mitochondrial functions. And this gives us a very comprehensive picture of the mitochondrial phenotype in a data set. So the pathways are divided up into levels. So you have high level pathways such as metabolism that have more specific functions within them. So metabolism contains citric TCA cycle and fatty acid oxidation, for example, as well as oxphos and the individual complexes. And then once we have that, we can then do downstream analyses to understand the mitotype in the data set that we're analyzing. So for example, we can use PCA to do clustering analysis to see if we have cell types or tissues with similar mitotypes or different mitotypes, as well as hierarchical clustering analyses to see which pathways are enriched. And then we can do further analyses looking at the top enriched pathways, like which pathways most distinguish the mitochondria between different tissues or cell types. And then we can use this to look into individual differences, such as how mitotypes shift with age, which I look at in my project. So the project I'm working on, I'm not working with MiSBIE data. I'm working on the MERC project, which stands for the Mitochondrial Immune RAN-CR project. So we were provided a data set by our collaborators. It's an immune cell pseudo-bulk RNA-seq PBMC data set. And it contains 164 women and men between the ages of 26 to 84, and I'm showing the age distribution there. So we have the full adult age distribution, which makes it ideal for investigating the effects of age on immune cell mitotypes. The data set contains 24 different immune cell types, which I'm showing here. So I have the hematopoietic stem and progenitor cells. I have the myeloid subtypes of dendritic cells, monocytes, NK cells, 9 different subtypes of T cells, including CD4, CD8, and then unconventional Ts, and then subtypes of B cells. And this is a flow chart illustrating the study. So we collected blood from the 164 people, isolated the PBMCs, and then our collaborators performed single cell sequencing and aggregated this data into pseudo-bulk to get cell type resolution transcriptomic data. And then I took this data set and performed our mitotyping analysis on it by filtering for the mitochondrial genes and quantifying the mitochondrial pathway specialization. And then I investigated the shifts that occur with age in immune cell mitotypes. So this is a principal component analysis based solely on the 149 mitochondria pathway scores. And you can see that with only these mitochondrial pathways, we can it's enough to distinguish or to separate the myeloid immune cells from the lymphoid immune cells. But yes, based only on mitochondrial pathways. And you can see also there's further clustering. The plot is colored by cell type, so there's further clustering by cell type in the plot. So this is all of the pathways that we investigated. So we actually ended up doing 151. We included glycolysis and pentose phosphate pathway, as they're important for determining the phenotype of immune cells. So this is to show the overall pattern. So here pathways are on the rows and the columns are cell types. And you can see that the cell types show a different, the mitochondrial, they show different mitochondrial profile in their expression patterns. So we performed hierarchical clustering on this and identified 6 broad mitotype clusters, including shown here in the radar charts, so including monocytes, naive and memory T cells, B cells, cytotoxic and innate lymphocytes, proliferating lymphocytes, and dendritic cells. But overall, the picture showed that immune cell types have distinct mitochondrial signatures based on their transcriptome. And then zooming into OXPHOS, we can see that it looks very different between different cell types. So here, red is high expression and blue is low expression. So we can see the monocytes on the left are very high prioritization of the OXPHOS pathway, as well. So are the B cells, which have even higher than the T cells. But some of the cells, such as the NK cells and dendritic cells, are quite low for OXPHOS, as well as the proliferating cell types. And then looking at glycolysis, we see a different signature. So the proliferating cells show a strong glycolysis signature, which is consistent with what we know about the metabolism of activated immune cells, which rely on, they ramp up glycolysis in order to fuel their proliferation. But monocytes also show a strong glycolysis signature, which is very interesting. And B cells showed the lowest glycolysis pathway score. So now, having these immune cell mitochondrial phenotypes, I next looked into the age-associated shifts that occur in the immune system. So to look at this, I'm showing here, so for every cell type where I had at least 100 people, I performed a Pearson R correlation with age for that pathway within each cell type. And red here is a positive correlation, and green is a negative correlation. So we saw cell type specific effects. So for example, on the leftmost pathway is glycolysis. In the T cells, we saw a strong positive correlation with age, particularly in Tregs, where it was significant after multiple testing, correcting for multiple testing. And also OXPHOS showed an interesting signature where some pathways, some cell types increased expression of OXPHOS with age, and then some showed a decrease. These are three representative plots that were found to be significant after multiple testing, correcting for multiple testing. So carnitine synthesis and transport in CD8 naive, glycolysis in Tregs, and then glyoxylate metabolism was down across most cell types, and especially so in gamma delta T cells. Overall, in this data set, it looked like lymphocytes were more sensitive to the effects of age than myeloid cells. So I'm showing here on the plot the number of significant pathways for each cell type. And we found particularly T cells seem to be the most affected by age. So I also, in addition to cell type specific signatures, I also saw immune-wide changes in mitochondrial pathway expression. So the pathways here that I have, the border around particularly lipid metabolism was up across most cell types, as well as detoxification and sulfur metabolism. And then we also saw some pathways that were deprioritized across most cell types. So the pathways related to central dogmat, which involved the maintenance of the mtDNA and also transcription and translation. So probably the ability to be able to perform mitochondrial biogenesis seemed to be deprioritized with age. So in order to look at this a bit more clearly, we averaged across each pathway across all immune cell types to look at which pathways were kind of showed this more high level immune-wide signature. So when we did this, fatty acid oxidation and carnitine shuttle and carnitine synthesis and transport were the most positively correlated, whereas mitochondrial ribosome assembly, iron sulfur biosynthesis, and pathways related to calcium homeostasis were deprioritized with age. So in conclusion, this data set, in this data set, we comprehensively profiled the mitochondrial phenotypes of immune cells at cell type resolution across the entire PBMC population. We found that immune cell subtypes display distinct transcriptome-based mitochondrial phenotypes, which we refer to as mitotypes, and also found that immune cell mitochondria undergo cell type specific and immune system wide pathway prioritization shifts with age. And we have a single cell RNA-seq PBMC dataset from the MiSBIE study, and we will be applying our mitotyping approach to analyze this data, and I think Anna's going to touch on that in the next talk. Thank you.